Okay, hi everyone. This is a lecture for Biochemistry 470, and the title of today's lecture is the Carbon Cycle Part 2. So for our lecture outline today, we are going to start by defining the global methane cycle. We'll also identify some anthropogenic sources of methane emissions and discuss methane as a greenhouse gas. And then we'll compare three biochemical pathways that are used for methane production, which is also known as methanogenesis. Okay, so as we can see here, the kind of chemical structure or chemical formula of methane is CH4. So kind of significantly, methane contains carbon in its most reduced form. Under most conditions, methane is a colorless, odorless gas, and it's significant in relation to the carbon cycle because methane is the second most abundant carbon-containing gas in the atmosphere. So if you look at this table here, which lists all of the gas constituents of the atmosphere, we can see that carbon dioxide is listed as having a concentration of 360 parts per million. This data is actually a little bit old. We know that the concentration of CO2 in the atmosphere is now about 410 parts per million. But kind of importantly here, we can see that the concentration of methane is much less. So listed here as 1.5 ppm. So more than 200-fold less than carbon dioxide. But it's still the second most abundant carbon-containing gas in the atmosphere. And so in relation to the carbon cycle, methane is a really significant gas. It's a significant gas because it's a greenhouse gas, and it actually has a higher global warming potential as compared to CO2. Okay, so we define the global methane cycle as being kind of a subcycle of the carbon cycle that describes the flux of methane between various reservoirs on Earth. And if we consider these reservoirs of methane, sort of the three most abundant methane reservoirs are ocean hydrates, permafrost hydrates, and gas reserves. So if we look over at this figure here on the left, which describes kind of the global methane cycle. So over here on the left, we can see that there are various processes that can function to remove methane from the atmosphere. We can have methane reacting with hydroxide radicals or chlorine in the atmosphere that functions to remove methane from the atmosphere. And in terrestrial environments, we can also have oxidation of methane by methanotrophic bacteria also functioning to remove methane from the atmosphere or from the air. And then sort of in the middle here, we can see a number of processes that function to emit methane. And kind of importantly, the arrows that are black indicate natural fluxes or natural processes, and the arrows that are red indicate sort of anthropogenic processes or fluxes. And then this arrow here that's kind of yellow or brown represents a process that's both anthropogenic as well as naturally driven. So in terms of methane emissions, we can see that we can have methane emitted from ocean hydrates. We also have a lot of methane emitted from agricultural processes, so methane emitted from livestock, as well as agricultural processes such as rice cultivation. We can have methane emitted from water supplies, so from freshwater or wetlands. Also, lots of methane emitted from kind of waste sources, so landfills and other sources of waste. We can also have methane emitted sort of during incomplete combustion of fossil fuels or kind of the burning of biomass. And so kind of importantly here, it's currently thought that about 50% of methane emissions are natural and about 50% of methane emissions are anthropogenic or kind of human driven. Okay, and so if we want to kind of consider methane emissions kind of from a more biological perspective, it's important to recognize that most methane emissions are driven by microbes or microbial processes. So this pie chart here on the left represents kind of total percentage of methane emissions or the total methane budget. And we can see here that sort of everything from kind of wetlands, which is purple, all the way down to sewage treatment here, which is kind of blue, everything sort of here. These are all microbial-driven processes. So about 70% of all methane emissions are produced by what are known as methanogenic archaea, or methanogens. And so we're going to talk a little bit more about these methanogenic archaea, or these methanogens that produce methane. So as I said, these organisms are known as methanogens. If we look at this first figure here, which shows kind of our phylogenetic tree of life, of course we have sort of three domains of life. We have bacteria, archaea, and eukarya. And methanogens sit within this kind of phylum of archaea on the right here, which are known as Euryarchaeota. So it's kind of this right branch here comprises these methanogens. So biological methane production is achieved by species of archaea that are known as methanogens. These methanogens are anaerobic, so they produce methane under anaerobic conditions. And they're also obligate methane producers, meaning that they have to produce methane to survive. There's also over 50 known species of methanogens. They can be either cocci or bacilli in shape. And this electron micrograph here is showing a culture of methanogens known as Methanobacterium formicicum, which as we can see here are bacilli. And kind of importantly, these methanogens are found in early diverse environments. So typically where we see methanogens sort of functioning ecologically is they tend to function sort of at the end of anaerobic food chains. So methanogens tend to thrive in environments where other nutrients, such as kind of sulfur or nitrogen, are limiting, and they can sort of function by, in anaerobic environments, kind of completing degradation of products produced by other bacteria, particularly kind of fermentation products that are produced by other bacteria or other microbes. And so we're going to sort of continue the lecture by covering sort of three pathways, the three primary pathways that are used by methanogens to produce methane or for methanogenesis. So the three pathways are shown here. The first one on the left is known as the hydrogenotrophic pathway. And in this case, methane is produced from carbon dioxide as well as hydrogen gas or molecular hydrogen. And in this case, CO2 is reduced to methane, and the electrons that are used to reduce CO2 to methane are derived from hydrogen. This central pathway here, which is shown in B, is known as the acetoclastic pathway. And in this case, methane is produced from acetate. So in this case, the methyl group within acetate is reduced to methane, and then this carboxyl group within acetate is oxidized to CO2. So we can produce methane from acetate within this central pathway. And then the final pathway, which is shown here on the right, or in C, is known as the methylotrophic pathway. And in this case, some of these archaea, or some of these methanogens, can also produce methane from methanol. So in this case, the methyl group within methanol is reduced to methane, and these archaea sometimes also oxidize this methyl group to CO2. And I'll talk about sort of these two branches of the pathway a little bit more later in the lecture. But sort of significantly, most methanogens use this hydrogenotrophic pathway. This is thought to be the primary pathway that these methanogens can use to produce methane. Okay, so kind of looking at these pathways in a little bit more detail. So the first one, this hydrogenotrophic pathway for methane production. So several steps within this pathway, and we'll kind of look at each of them individually. So the first step in this pathway is reduction of ferredoxin by molecular hydrogen. And ferredoxin is an electron carrier. It's used by lots of anaerobic organisms, anaerobic bacteria or archaea, as well as a lot of photosynthetic organisms. So we first have reduction of ferredoxin by molecular hydrogen. And then the second step, which is shown right here, is known as reductive activation of carbon dioxide to form formyl methanofuran. So in this case, we have reduction of CO2 to a formyl group. And it's also called reductive activation because that formyl group is also conjugated to a coenzyme, which is known as methanofuran. And so we can sort of look at methanofuran in a little bit more detail here. So at the top, this is the structure of this coenzyme methanofuran. This part over here is 2-aminomethylfuran, and then it's 2-aminomethylfuran, sorry, here, and then it's conjugated to a phenoxy group. Not terribly important the structure of this thing, but significantly this amino group here is kind of the functional group where the formal group is conjugated. So if we look down here at this representation, we can see we have CO2 as well as our methanofuran, and then here's our formal group attached right here to the coenzyme. And the dehydrogenase that catalyzes this reaction, pretty significant enzyme in methanogenesis, and it has sort of two active sites, and we can kind of see this diagram of this enzyme functioning here. So electrons from ferredoxin are kind of funneled into this first active site here, which contains tungsten, and then CO2 is reduced to formate in this first active site here. And then down in this second active site, which contains zinc, is where we have conjugation of the formal group to methanofuran to form formal methanofuran down here, this dehydrogenase. Okay, so kind of moving on with the pathway then, the third step in this pathway involves the transfer of this formal group from methanofuran to another coenzyme, which is known as H4-MPT, or tetrahydromethanopterin. So 3, we have the transfer of this formal group from methanofuran to H4-MPT. And then in step 4, we have conversion of the formal group to a methanol group, and this is catalyzed by a cyclohydrolase. And we'll kind of look a little bit more detail at the structure of this coenzyme H4-MPT as well. So this is a chemical structure of tetrahydromethanopterin. It has a really similar chemical structure to tetrahydrofolate, which is probably a coenzyme that a lot of you are more familiar with. And actually the methyl groups that are shown in blue here kind of indicate how the structure of the other coenzyme, tetrahydrofolate, sorry. And sort of significant in relation to this coenzyme, we have sort of the primary kind of active sites, or kind of the primary conjugation sites are kind of this N5 and this N10 site here within the coenzyme. So we can see if this is kind of a simplified structure of the coenzyme, we can see first the attachment of the formal group first is attached to this N5 site, and then through the function of this cyclohydrolase, we form this methanol H4-MPT. And then subsequently in the pathway, which we'll see in a minute, this methanol H4-MPT is reduced to a methylene H4-MPT, and then reduced again to a methyl H4-MPT. And here's our methyl group here, which is attached to this N5 site within the coenzyme. Okay, so kind of moving on to steps 5 and 6. So step 5, as I kind of just said, is reduction of the methanol group to a methylene group within this, still bound to this coenzyme H4-MPT. Step 6 is then reduction of this methylene group to a methyl group. And so here we have a methyl group attached to H4-MPT. And both of these reduction reactions are catalyzed by dehydrogenases, which use a coenzyme which is known as F420. So we can quickly kind of look at the structure of this coenzyme F420 as well. Kind of interestingly, this coenzyme F420 is a flavin derivative, meaning that it's derived from riboflavin, which is a B vitamin. It's chemically or structurally similar to coenzymes such as flavin mononucleotide or flavin adenine dinucleotide, which some of you have seen obviously before in previous courses. But kind of, I guess, significantly, this is the oxidized form of the coenzyme as well as the reduced form of this coenzyme F420, which is used in these reduction reactions. Okay, and then the final 3 steps of this pathway, 7, 8, and 9, is where we actually see methane production. So in step 7, we have transfer of the methyl group from H4-MPT to this coenzyme known as coenzyme M. And then in step 8, we have reduction of the methyl group within coenzyme M, or methyl coenzyme M, reduction of this methyl group to methane by coenzyme B. And this also forms this mixed disulfide, which is shown here. And then the final step in the reaction is a final reduction reaction that reduces this mixed disulfide back to regenerate the reduced forms of this coenzyme M and coenzyme B here. So this is known as the hydrogenotrophic pathway. And I guess we can look just a little bit at sort of the chemical structure of these coenzyme M and coenzyme B as well. So importantly, here's methyl coenzyme M, here's our methyl group, as well as coenzyme B. Both of them contain a sulfhydryl or a thiol group. And so we have reduction of coenzyme M by coenzyme B, forming this mixed disulfide here, as well as methane, which is the product of this hydrogenotrophic pathway. Okay, so the second two methanogenesis pathways are simpler to cover because they're kind of derivatives of this initial hydrogenotrophic pathway. So the second one is known as the acetoclastic pathway. And in this case, we have formation of methane from acetate. So the first step in this pathway is formation of acetyl-CoA from acetate. So here's acetate and the formation of acetyl-CoA. This is catalyzed by acetyl-CoA synthetase. And then the second step in this reaction is forming methyl H4-MPT. And this involves a transfer of a methyl group from acetyl-CoA to H4-MPT, as well as oxidation of this carbon, which was our carboxyl group here within acetate, to carbon dioxide. And then the last three steps of this pathway, sort of steps 3 and 4 and 5, are really the same as the hydrogenotrophic pathway, where we have a transfer of this methyl group to coenzyme M, reduction of coenzyme M by coenzyme B, forming methane, as well as our mixed disulfide, and then reduction of the mixed disulfide to regenerate coenzyme M and coenzyme B by some sort of reducing agent here. So this is known as the acetoclastic pathway, where we can produce methane from acetate. And then the final pathway for methanogenesis or methane production is the methylotropic pathway. So in this case, we're producing methane from methanol, as well as sort of similar C1 compounds. So in this case, we have the methyl group within methanol. Through the first reaction is transferred to coenzyme M. And this is catalyzed by some sort of methyl transferase. And if this is a C1 compound other than methanol, then it's a different sort of methyl transferase that can then transfer this methyl group to feed into this pathway. And then the bottom section of this pathway is really the same as the acetoclastic pathway as well as the hydrogenotrophic pathway. So we have reduction of this methyl coenzyme M by coenzyme B, forming methane. And then the final, as well as this mixed disulfide, and then the final step is reducing this mixed disulfide to kind of regenerate these two coenzymes. But this bottom section of this pathway consumes 2 electrons to reduce the mixed disulfide. And so these bacteria run sort of a combination of the bottom section of this pathway as well as the top portion of this pathway, which we'll look at here. And the top portion of this pathway, which oxidizes a methyl group to CO2, is really just the reverse of the hydrogenotrophic pathway. So in this case, we have a transfer of the methyl group to coenzyme M, and then oxidation of this methyl group to carbon dioxide. And this sequence of reactions here releases 6 electrons. And so these archaea, or these methanogens, can sort of produce methane as well as CO2 at a stoichiometry of sort of 3:1 to kind of release electrons to fuel this reduction here. So they produce both methane and CO2 through this methylotropic pathway. Okay, so I think now that we've kind of looked at these 3 biochemical pathways for methane production, I think the sort of central question here, or the thing that we want to sort of resolve is well, why do these methanogens produce methane? And the answer is that methanogenesis comprises a variation or a type of anaerobic respiration. So it's a means that these archaea, or these methanogens, can use to generate energy, or to generate a gradient across a membrane to produce ATP. So it's a version of anaerobic respiration. Methanogenesis is also thought to be a lot more diverse and complex and varied than most other types of respiration, such as oxidative phosphorylation or photosynthesis, or some other types of anaerobic respiration. So we'll just look at one example here where we can see methanogenesis functioning as an electron transport chain. Okay, so if we look at this diagram here, we can see that we've got sort of a similar representation of this hydrogenotropic pathway, but we can see that it's functioning as this electron transport chain. So if we have a CO2 kind of outside of these archaea, diffusing across the cytoplasmic membrane into the cytoplasm, we can then have CO2 being reduced to methyl coenzyme M here in the cytosol. And kind of importantly, the second step of this process, so the formation of formal methanofuran, consumes 2 protons within the cytoplasm. So then the second step of this process then would be reduction of this methyl coenzyme M by coenzyme B to form the mixed disulfide here in the cytoplasm. And then now we can kind of see where this electron transport chain sort of comes into play. So if we have oxidation of molecular hydrogen by a hydrogenase that's sort of membrane bound or transmembrane, producing 2 protons outside of the archaea, we can have 2 electrons then transferred through this electron transport chain. So transferred through cytochromes, as well as MPH2, which is methanophenazine, which is a hydrophobic electron carrier that functions within these methanogens. And then, so we can say that electrons are transferred between cytochromes as well as MPH2 to the reductase here. And then this reductase functions to reduce the heterodisulfide, or the mixed disulfide, to regenerate coenzyme B and coenzyme M. And so significantly, this system functions as an electron transport chain to generate a proton gradient across this cytoplasmic membrane. And so in this case, we had 2 protons consumed to form formal methanofuran. 2 protons are also consumed to reduce MPH2 and then also released into the extracellular space or outside of the bacteria. And we also had 2 protons generated here through oxidation of molecular hydrogen by this hydrogenase, resulting in this proton gradient across the cytoplasmic membrane. Okay, so I think what we want to do is just finish off the lecture by sort of reminding ourselves that the reason we're discussing the global methane cycle is because both carbon dioxide and methane function as very significant greenhouse gases. So as we can see on the top sort of graph here, atmospheric concentrations of carbon dioxide have increased by almost 50% since pre-industrial times. So if we look at sort of pre-industrial times, so maybe the year 1750 here, carbon dioxide levels increased from about 280 parts per million up into around 410 or 410 parts per million today. And they've been increasing at a rate of approximately 2 parts per million per year. And if we consider methane, you can see that in regards to methane, kind of that pre-industrial times here, sort of 1750, methane was present in the atmosphere at around 720 parts per billion. And it's increased by more than 150%. The concentration of methane in the atmosphere now is about 1900 parts per billion. And so concentrations of methane are increasing at about 8 parts per, this should be parts per billion per year. And so although concentrations of methane are much lower in the atmosphere as compared to carbon dioxide, methane has a much higher global warming potential as compared to CO2. And so it's thought that currently the overall contribution of methane to the greenhouse effect is somewhere in the range of kind of 10 to 15%. So a really significant greenhouse gas. Okay, so just to finish this lecture, The global methane cycle describes the flux of methane between various reservoirs on Earth. Methanogenic archaea, also known as methanogens, account for 70% of global methane emissions. The hydrogenotrophic, acetoclastic, and methylotrophic pathways are the biochemical pathways that are used to produce methane. And atmospheric methane concentrations have increased 2.5-fold since pre-industrial times, and methane is considered the second most significant anthropogenic greenhouse gas. Okay, thanks for watching. Bye.